At present, there are only three essential ways of treating cancer: attacking its growth by chemotherapy and/or radiotherapy, blocking hormones that stimulate its growth, as with breast and prostate cancer, enhancing the body's natural immune rejection of cancer. I propose a potential fourth way: attacking cancer by depriving it of energy. 2 years ago, at the outset of the 21st century, there were many ideas about the future. But at that moment, I was still struggling to answer the many questions that have been floating around my brain from earlier years. These questions constitute the background to what I will be attempting to explain today. Cancer, often called the Big C by many of my patients, can hit us like a car crash. Its brakes suppressor genes don't work, and the accelerator oncogenes is jammed on. When I started as a young oncologist in 1970, the multiple disparate carcinogenic factors obscured the central fact that cancer was simply the result of genetic mutation. Today, the somatic mutation theory of cancer is widely accepted. Mutations in the on and off switches controlling cell division can transform normal cells into cancer cells that divide relentlessly. Growing in tissue culture flasks under optimal conditions, these cells form colonies. In my laboratory in the 1980s, we were measuring the effect of chemotherapy by its ability to inhibit the growth of cancer cells into colonies. We worked with adherent cell lines and counted the flat colonies that grew over 10 days and were then fixed and stained with Giemsa. To compare different colonies from different cell lines, the number of colonies in the treated wells were divided by the number of cells in the control wells and adjusted to 100%. When I was counting colonies, however, I noticed that often on the control plate there would be failed colonies which had spontaneously died, even after many generations. At the top of this slide, two examples of failed colonies, each to the right of intact colonies, are shown for human colorectal and non-small cell lung cancer. To investigate spontaneous death of untreated cancer cells, my technician Ros White counted the percentage of untreated control cells which formed colonies in 18 human cancer cell lines, bottom left. Only between 30 and 80% of untreated control cells formed colonies. The histogram, bottom right, compares the potential doubling times measured by flow cytometry with the actual doubling times observed by counting the number of live cells per flask in 8 human cancer cell lines. The actual doubling times were always lower than the potential doubling times. I needed to revise my perception of cancer cells, from the widely accepted concept of supercells to that of badly damaged zombie cells just managing to survive. But why was spontaneous death a hallmark of highly selected cancer cells growing under optimal tissue culture conditions in the laboratory? A key experiment by Gerard Evan at the end of the last millennium gave me a clue. Gerard demonstrated that oncogenic transformation by the MYC oncogene of normal mouse embryonic fibroblasts could result in apoptosis as well as cellular proliferation. In order to survive, the oncogene transformed cell must avoid apoptosis. Several methods of avoiding apoptosis have been described in well-established cancer cell lines in the literature. But how does a normal cell, at the first instant of oncogenic transformation, avoid apoptosis? The clue at the bottom of the list lay in the fact that apoptosis is an energy-dependent process. Cancer cells at the moment of oncogenic transformation require levels of ATP to be below that which allows apoptosis to occur. In cancer cells, ATP is produced by aerobic glycolysis. Nearly 100 years ago, Otto Friedrich Warburg observed that cancer cells have different modes of metabolism from normal cells. They were not using mitochondrial oxidative phosphorylation, but have switched to only using glycolysis, despite being fully oxygenated. The aerobic glycolysis switch, shown at the top of the slide, is at the end of a long glycolytic chain, shown below. At the aerobic glycolysis switch, the phosphoenol pyruvate is converted to pyruvate with the production of one molecule of ATP. In cancer cells, this is carried out by the enzyme PKM2. The pyruvate created by PKM2 does not enter the mitochondria, but is converted to lactate with the oxidation of NADH to NAD. To compensate for the small amount of ATP produced per glucose molecule by aerobic glycolysis, glucose uptake across the cell membrane is greatly increased, as Warburg first observed. We see the evidence of this in the detection of cancer by PET scans. The positron emission of fluorine-18 annihilates by hitting an electron, creating two 511 keV gamma rays at 180 degrees for the ring detector. The reason the technique identifies cancer, however, is because the fluorine isotope is attached to deoxyglucose, which has an increased uptake across the cancer cell membrane and then is trapped intracellularly, because after phosphorylation by hexokinase, it cannot be further metabolized by the enzyme phosphoglucose isomerase. The progressive accumulation of 18F-deoxyglucose shows as a signal on the PET scan. Warburg's early observation has now been given a molecular mechanism. Aerobic glycolysis is the result of a switch from expression of exon 9 to exon 10 of the PKM gene. PKM2 can function as a dimer with multiple interaction sites and interaction factors. It is a PKM tetramer which is responsible for the release of ATP during the conversion of phosphoenolpyruvate to pyruvate. At present, only two factors are known to affect tetramerization of PKM2. 1,3-fructose biphosphate, produced earlier in glycolysis, binds at the tetrameric interface and promotes its activity by inducing allosteric changes. Also, serine has been shown to bind and activate tetrameric PKM2. How serine affects the rate of glycolysis and relates to FBP-induced allosteric changes is not known. There are no known therapeutic drugs that interact with PKM2 at its tetramerization interface. But why is PKM2 expression a hallmark of cancer cells? I find it hard to believe a whole variety of normal cells undergo malignant transformation and then decide for some inexplicable reason to adopt aerobic glycolysis. I suggest that cancer starts in PKM2 cells for reasons that will emerge as the talk progresses. In support of this hypothesis, Warburg-like changes have been observed in adjacent normal regional tissues in cervical and colorectal cancer. This poses the question, is PKM2 expression an important change of pre-malignancy? I realized early on in my career as a clinical oncologist that every patient was an individual and every cancer was unique. No two cancers, even if they were classified as being of the same histological type, necessarily behaved in the same way in the clinic. DNA sequencing is now caught up. I think finally is in agreement with what I intuitively believed to be true years ago. When I look at the pathology slide of cancer our team was treating, it was clear that the cancer could only be described as chaotic in comparison to normal tissues. As the closest in vitro model of this, my laboratory chose to examine relative phenotypic expression of cancer-related proteins across a wide range of different human cancer cell lines and primary cultures of human diploid fibroblasts and keratinocytes. My PhD student, Lawrence Zebra, used semi-quantitative Western blotting to measure the relative expression of 18 key proteins involved in control of proliferation and death in 20 human cancer cell lines. Also, with Lita Carizzi, he measured these same proteins across 8 individual primary cultures of human diploid keratinocytes. Homogeneity of proteomic expression was tested by demonstrating positive pairwise cross-correlations. In the case of normal keratinocytes, there were several highly significant pairwise cross-correlations, shown in green. But there were only 3 cross-correlations of low significance found in the 20 human cancer cell lines, shown in red, consistent with marked cancer heterogeneity. The only strongly positive correlations in the cancer cells was the surprising co-relationship between CDK4 and CDK1, shown in the smaller upper figure in red. By contrast, in normal keratinocytes, CDK4 and CDK1 in green showed a slight negative correlation. Lawrence Zebra explored the CDK4-CDK1 positive relationship by transfecting human 27 ovarian cancer cells with wild-type CDK4 in a myristoyl-inducible vector, constructed by Shanez Anandappa and Rasheen Dormer. The results are shown here. 1 millimolar myristoyl induced both CDK4 and CDK1 proteins, with no time delay between the two, shown in panel A. I noted that unexpectedly, there was no phosphorylation of the retinoblastoma protein associated with CDK1 elevation caused by myristoyl-induced CDK4, panel B, top right. This observation caused me to wonder if the CDK4 protein might also have a non-kinase function. If so, I expected it might be located in a site distant from the kinase region itself. I searched the amino acid sequence of CDK4 and noted that the kinase region and all the known functional regions lay in the N-terminal 2/3 to the left, panel C. I questioned whether there might be a non-kinase functional site in the C-terminal region, such as the NKR site shown in blue in panel C at the bottom on the right. I asked Professor Jeremy Kilburn at Southampton University if he could perform a structural study to see if there was something different in the region I suspected. He asked me what exactly in chemical terms I meant by something different, but nevertheless carried out a structural study to compare the C-terminal region of human CDK4 with the highly conserved, closely related proteins CDK2 and CDK6. When we met 2 to 3 months later, Jeremy immediately said, "Hilmar, I think I have found something different." This slide shows the linear amino acid sequences of the C-terminal regions of CDK4, CDK6, and CDK2, aligned in descending order. First, I'd like to draw your attention to the red and blue boxes, which show homology and partial homology respectively. This can be seen between the C-terminal regions of CDK6 and CDK2, but there's no homology between either of these and the C-terminal amino acid sequence of the very closely related peptide CDK4. Secondly, in black and yellow, depicted in bold capitals, in the decapeptide is the decapeptide sequence FP, PRG, PRP, VQ, which is uniquely present in the C-terminus of CDK4. Classically, CDK4 works as a kinase, which phosphorylates the retinoblastoma protein after forming a Holder enzyme with cyclin D. However, a knockdown of CDK4 gene alone in mice has revealed that the normal CDK4 protein can independently have a critical role in carcinogenesis without the need of its cyclin D partner. Knockdown of CDK4 completely prevents carcinogen-induced skin cancer in mice, despite the continuing presence of CDK2 and CDK6, and without affecting normal keratinocyte proliferation. CDK4 knockdown blocks c-Myc-induced skin cancer in K5-Myc mice and also stops RAS transformation of dominant negative p53 or ARF-INC4 null mouse embryonic fibroblasts. In addition, it delays the onset and lowers the incidence of mammary carcinoma in MMTV-new null mice. The central amino acid sequence, PRG-PRP, of the previously unrecognized decapeptide was prepared by Dr. John Smith in Professor Philip Rudland's biochemistry department using an automated peptide synthesizer. I tested the PRG-PRP hexapeptide for biological activity by adding it to tissue culture wells containing either RT112 bladder cancer cells or normal diploid human fibroblasts. Over the following week, I personally observed the cells in the tissue culture wells closely. The RT112 cancer cells seemed to be stimulated rather than suppressed, causing a lot of joking by my fellow laboratory workers. At that stage, it was time for me to go on annual leave. When I returned 2 weeks later, my chief technician, Carol Thomas, said, "You ought to take a look at the cells you were working with 3 weeks ago. They're still in the incubator." In my not unusual forgetful manner, I had left the multi-well plate I'd started testing earlier in the back of the incubator. Here is the low-power photomicrograph I took 20 years ago of the cells in the multi-wells of the plate left at the back of the incubator. To my absolute amazement, the PRG-PRP treated RT112 bladder cancer cells, top right, were all dead. The fibroblasts were growing happily in the presence of PRG-PRP, as were the control untreated RT112 bladder cancer cells, top left. I simply could not believe that a simple linear peptide of 6 normal amino acids in the same sequence as those in the C-terminus of wild-type CDK4 were able to selectively kill cancer cells. I tested a variety of different versions of the linear hexapeptide sequence on several of the many different cancer cell lines growing in my laboratory, but only PRG-PRP was active. Changing the non-arginine amino acids had little effect, but changing PRG-PRP to PRR-PGP completely removed the cell-killing effect. The relative positions of the arginines was key to function. I was concerned, however, that I was using very unphysiologically high concentrations in the millimolar range to cause cancer cell death. I needed to increase the concentration of the peptide. To improve cellular uptake, I concentrated a cyclic version of the PRGP warhead in an amphiphilic cassette. PRG-PRP is itself amphiphilic. This was designated THR-53 after Ferrite, the company I founded with the approval of the University of Liverpool. THR-53 had a 10-fold greater activity than the linear PRG-PRP peptide, with an LD50 of 200 micromolar. The majority of our cancer cell lines were of epithelial origin, and so the effect of PRG-PRP was tested on short-term colonies of normal human diploid keratinocytes, panel A, which, like human fibroblasts, were not affected by the peptide. THR-53C had a PRR-PGP warhead, as with the linear peptide, panel B1. The altered relative positions of the arginines in THR-53C completely removed the cancer cell cytotoxicity, panel B2. Finally, THR-53 was tested on around a dozen different cell lines that had originally been explanted from different tumors in different patients in different parts of the world. All different types of cancer appeared to die in the same way, as shown by the 4 typical examples in panel C. I still find it hard to believe that I'm describing to you in Lisbon a global cancer killing effect which spares normal cells. Although based on many different cell lines, this is of course only in vitro data. At that time, after leaving Liverpool University, I had no personal laboratory. The experimental results shown on the previous slide were commissioned by my private consultancy company, Hillross, which ordered custom peptides personally designed by me from PepScan, a company in the Netherlands. These were then sent direct to Horizon Discoveries, a company in Cambridge, UK, for biological testing. I was not, therefore, directly involved personally in the actual experiments. So I'm forced to believe the results shown here, which I published with my colleagues in 2011, are in fact reliable. The cyclic amphiphilic peptide THR-53, carrying the PRG-PRP amino acid sequence in its warhead, selectively kills cancer cells by necrosis. The left-hand panel compares trypan blue viability following THR-53 in the non-malignant immortalized cell line MRC5H TERT. Below is the MCF7 human breast cancer cell line. For MRC5H TERT, the solid red line indicating viable cells following treatment rises at the same rate as untreated control cells, while there are a few dead cells at the bottom. Underneath, it can be seen that the opposite happens in the case of MCF7 breast cancer cells. The broken red line indicating dead treated cells rises at the same rate as the control cells over 7 days, while the solid red line of viable treated cancer cells does not increase. Identical results were obtained with H460 and H299 human non-small cell lung cancers. The morphology of dying dead H460 cells is shown top right. The dying cells are fragmenting. No signs of autophagy or apoptosis can be seen. The flow cytometry histogram below is typical of those that were seen throughout PRG-PRP treatment, with no evidence of any change in cell cycle distribution and no sign of a subdiploid peak that would indicate apoptosis. I've been unable to understand why PRG-PRP was causing colonies of cancer cells to all die in the same way, with the distinctive morphology shown on this slide. I was at a meeting in Harnia on Crete, and had just finished describing these findings at a sparsely attended early morning small group meeting. As I left, a lady, I believe she might have been Italian, stopped me by the exit and said, "I know why your cells are dying. I've seen it before. They're running out of ATP. I've seen this appearance many times." This was a really helpful clue. A graph of ATP levels in the first days after addition of THR53, assayed by cell T to glow luciferase assay, at the top of this slide, shows THR53 causes an abrupt drop in ATP without there being any effect on mitochondrial function measured by Alamar blue or cellular protein levels as measured by sulfur-rhodamine B below. Pharmacokinetic studies in mice were carried out on THR53 with the intention of progressing to test it on human tumor xenografts. It was cleared very rapidly from the blood after a single intravenous injection, however, and because it's also had a low specific activity, it could never achieve high therapeutic levels. Therefore, I personally designed a succession of modifications to THR53 over 15 years. These were commissioned from PepScan in the Netherlands and sent to Horizon Discoveries for biological testing. Combining the optimal modifications yielded HILR 056, which I call ONKONEK. The improvement in the LD50 from PRG-PRP to HILR 056 was of the order of 5,000. Each of the new peptides I commissioned was increasingly hydrophobic, making it necessary to use the proprietary nanoparticle-based delivery system Nanosyn from Tecrea Limited for efficient solubilization in vitro. In parallel with developing a therapeutic drug, I continue to seek a potential mechanism to explain how a simple 6-amino acid sequence from a non-kinase region of wild-type normal CDK4 could globally selectively kill cancer cells by depriving them of ATP without having any effect on cell division. I'd found an interesting jigsaw piece, but I'd no idea which jigsaw it fitted or where to locate that particular jigsaw. I sought potential mechanisms in the literature which could give rise to necrosis as a result of low ATP levels. A paper by El-Bashir Athar described how poly-ATP ribose polymerase, PARP, could cause necrosis by using up NAD and depleting ATP. So I tested the effect of the PARP inhibitor olaparib on PRG-PRP killing of cells in vivo. The necrosis caused by PRG-PRP was completely reversed by olaparib, as was the fall in ATP levels, as shown in the top panel. I thought that possibly PRG-PRP could be a PARP agonist, but comparison of the two haploid lines, in one of which PARP had been knocked out, produced two completely overlying cell survival curves shown below. Thus, my idea that PRG-PRP might be working as a PARP agonist was incorrect. However, the PRGP warhead in the cyclic amphiphilic peptide HILR 025 at a dose close to the LD50 for necrosis caused inhibition of lactate dehydrogenase, which is often used as a downstream indicator of pyruvate kinase activity. It seemed that PRGP might be inhibiting aerobic glycolysis rather than augmenting PARP. In an attempt to discover an aerobic glycolysis site, which the hydrophobic cationic PRG-PRP region might potentially interact, I searched the linear amino acid sequences of the key aerobic glycolysis enzymes phosphofructokinase, hexokinase, lactate dehydrogenase, and pyruvate kinase 2. The sequence I was seeking should have two anionic amino acids at the same distance apart as the cationic arginines of PRG-PRP, and like them, lie in a nest of hydrophobic amino acids. There were a number of potential sites in LDH, but I could not imagine how PRG-PRP could produce a drop in ATP levels by interacting with LDH. Otherwise, It was only at the C-terminal end of the amino acid sequence unique to PMK2 and to cancer, underlined in red on the left of this slide, lying between amino acids 378 and 411 and expressed by exon 10 of the pyruvate kinase gene, that I discovered the amino acid sequence SDP-TEA. This diagram shows a suggested alignment of PRG-PRP and its putative target SDP-TEA. The anionic aspartic acid and glutamic acid residues in a hydrophobic background of nonpolar amino acids could potentially align with the cationic arginines of PRG-PRP, also in a hydrophobic background. The PKM1 amino acid sequence in this site is TDL-MEA. PRR-PGP, previously shown to be unable to induce necrosis as a linear peptide or in the warhead of THR53C, does not align with SDP-TEA. I suggest that PRG-PRP within the C-terminal region of CDK4 or as a warhead in a cyclic amphiphilic drug may target the SDP-TEA peptide sequence within the tetrameric interface unique to PKM2. Based on the present evidence, I propose the hypothesis that cancer arises in normal cells that have switched to expressing the PKM2 isotype. PKM2 expression in normal cells is probably a characteristic of premalignancy. At the initial instant of transformation from a normal cell to a cancer cell, a somatic oncogenic mutation switches on cell mutation by upregulating CDK4, as well as initiating DNA synthesis by its well-known kinase activity. Upregulated CDK4 also downregulates PKM2, so that there is insufficient ATP to allow apoptosis to occur. My hypothesis suggesting what might happen at the initial moment of oncogenesis of a normal cell to a cancer cell is consistent with the proposed mechanism for the production of necrosis by PRG-PRP and the related molecules such as HRO56. On this slide, the panel on the left shows how the oncogene-transformed cancer cell could avoid apoptosis because CDK4 interacts with PKM2 to downregulate ATP to below the threshold for apoptosis, but not so low that the cell is unable to survive. I believe I've managed to hijack this mechanism by synthesizing a hexapeptide homologous to the PRGP amino acid sequence in the NKR region of CDK4, and then developed increasingly potent cyclic amphiphilic peptides containing PRG-PRP or highly polar variants of PRG-PRP, with unnatural amino acids surrounding the two key arginines. The final unique congener is HRO56, or ONKONEK, shown in the right-hand panel. The specific activity of ONKONEK against PKM2 causes too great a drop in ATP. This results in failure of the cell membrane pumps because of insufficient energy, causing the cell effectively to drown. At present, I'm still only able to present a hypothesis, which, though it appears to make sense, requires much more detailed experimental confirmation. I'm working on an assay to show that CDK4 downregulates ATP production by interfering with tetramerization of PKM2. This assay can be used to compare the effects of normal CDK4 with CDK4 construct with a site-directed mutation that expresses PRR-PGP rather than PRG-PRP. Modeling studies are also planned to test the hypothesis that PRG-PRP physically interacts with SDP-TEA. I do not believe this to be a tight, irreversible binding. To work as a PKM2 modulator, the binding would need to be weak and potentially reversible. Our early experiments have shown that PRG-PRP only causes a drop in ATP while it's present in the medium. Because of this, and in vitro evidence of lack of normal tissue toxicity, animal studies with ONKONEK, which are about to be carried out, are exploring the use of continuous infusion osmotic pumps rather than single subdermal or intravenous injections. I'd like to express my gratitude for the tremendous help many people have given me over the past 20 years. I am extremely grateful to a small local charity called Cancer and Polio Research Fund, based on Wirral in the north-west of England, and for the encouragement of its founder, J.K. Douglas, and his grandson, Andrew Douglas, who supported my work at Liverpool University when larger national challenges showed little interest. My first small biotech company, Therite, supported the structural chemistry at Southampton. Over the past 17 years, I have vacated my position and my lab at the University of Liverpool and have funded from bank loans and equity released on my house research by the commercial companies PepScan, now Biosynth in the Netherlands, Horizon Discoveries based in Cambridge, UK, and over the past 3 years, Celematics based in Nottingham, UK. Listed on the right are the many scientists who've worked on the program, several in my laboratory and others in collaborating departments. The one key name missing on this list is that of the extremely helpful lady on the meeting in Crete, who gave me the key clue about ATP, and who sadly I've never had the opportunity to meet again. Wherever she may be, I send my profound thanks. Thank you.